Hello, hi, a very happy good evening. So this is Krishnaveni. We'll be talking about the story of life on Earth, and that's biology. So welcome to my lecture today. So today we're just going to discuss the important questions from the chapter Cell Cycle and Cell Division. So we have recently covered this chapter. So today I'm going to discuss some important questions, some tricky questions where as a student you will have a doubt. So that's what the questions that I'm going to discuss. So please join me for this session. So please do like, share and subscribe. So share the link with your friends. So one share, that one share option that you use, so use it very carefully and use it very widely also, so that your friends also know here we are solving a lot of questions. All right, yes. So here we go, important questions from the chapter Cell Cycle and Cell Division. So if you ask me, ma'am, is this chapter tough? It is not tough, but yes, questions can be tricky. You might fail to understand the exact logic that is present in the question, so you might go wrong in just an instant. So in 2 seconds, if you have a wrong understanding of the question and if you have a wrong understanding of the concept, then everything will put into a wrong combination, so thus you will lose 4 marks for a good question. Alright, so thus to avoid all that, so here we are discussing important questions. So before we go forward, yes, Dharani, yes, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Okay, so before we go forward with today's question, so I have an announcement to make. So tomorrow at 4 p.m. we are starting human physiology. So I'm starting with the first chapter, that is digestion and absorption. So please don't forget to join me for the same. And tomorrow at 6:30 p.m. so we have guess the mentee chapter. So it is episode 9 tomorrow. So please stay tuned to find out what the chapter is all about. Okay, yes. So thank you once again for joining my lecture. So shall we move forward with the important questions? Yes. Okay, so here we move forward. So what are the topics that we discuss in this chapter, Cell Cycle and Cell Division? So it's going to be a very short lecture, so I have no big questions. The chapter itself is small and it has a weightage of just 2%. So I'm not going to take a lot of questions, just a small bit of questions. But yes, these questions can be a bit confusing. Okay, yes. Okay, yes, I will share the link with you. Okay, Dharani? So I was busy after that class. So now after this class, I will share the link with you. Okay, Dharani? Yes? Okay, so this chapter has no tough questions, not so easy questions as well. But let's see what kind of questions are there. So I have only limited set of questions. So let's see how those questions are described. So what are the topics that we were discussing in this chapter? So we were talking about, or we have defined the term, what is cell cycle, right? So that was the most important task. So first we discussed or we defined what a cell cycle is. So then we spoke about the phases of your cell cycle. So we have your interphase, you have your G1 phase, you have your G2 phase, then you have your S phase as well. So you have your mitotic phase. So then you have meiosis. So we were talking about all that. So who regulates your cell cycle? So that is an important question, right? So who regulates your cell cycle? So that was the important question of the hour. Then we spoke about mitosis, and then we also spoke about significance of mitosis. So moving further, we went on to talk about your meiosis. So we spoke about meiosis 1 and meiosis 2, right? Okay. So moving forward, giving you a quick recap of the important points in this chapter. So we defined what your cell cycle is. So what is a cell cycle? The sequence of events which includes your cell growth, your DNA replication, as well as your cell division is known as your cell cycle. Right? So your cell cycle has 4 phases. Yes, Shimpi, hello, a very happy good evening. So yes, I'm good, thank you. How are you? So thank you so much for joining my lecture today. So please do like, share and subscribe. Yes, Shubham sir, hello, very happy good evening. So thank you for joining us this evening. So please do like, share and subscribe. Okay, so this is about your cell cycle. So now what are the phases of your cell cycle? So your cell cycle starts with your G1 phase. Then we move on to the S phase. Then you have your G2 phase, and then you have your M phase, right? Okay, so this is basically divided into 2 phases. The one is known as your interphase and the other is your actual division, that is mitotic phase, right? So this is G1, S and G2. So this is known as interphase or the resting phase, but remember here your cell is metabolically very active, okay? So ma'am, if I start preparation from now, can I crack NEET 2022? Yes, aram se you can do. So you have hell a lot of time. So you can clear not, you can get 700 out of 720 out of 720. So you can do wonders, provided if you do it with a constant determination. Okay, Satya? Yes. So anyways, thank you Satya for joining my lecture today. So please do like, share and subscribe. So these are the phases, right? So who regulates your cell cycle? Who regulates them? So you have your cyclins. So you have your G1 cyclin. So you have G1S. So you have S cyclin and then you have M cyclins, right? So these are the different types of cyclins. So cyclins alone cannot regulate. So you have cyclin dependent kinases. So kinases are proteins which phosphorylate a particular substrate, right? So these two has to pair with each other or partner with each other so that they can regulate your cell cycle. Okay, so then we went on to talk about each of them in detail. So your interphase is where your cell is metabolically active and it is the longest phase of your cell cycle. So this involves your G1 phase, your S phase and your G2 phase. So in your G1 phase, this is the most variable phase your cell grows in this phase. Why it is variable phase is because here your cells decide whether they have to actually undergo division or not. So if they do not have to undergo division, they enter into a resting phase, that is G0 phase. So this is known as your quiescent or your dormant phase. All right. How to make short notes of biology? Okay, so this is how you have to make. So I have recently taught this chapter, okay. Now I'm going to discuss questions, but directly I'm not starting with the questions. I'm giving a short recap. So whatever I have discussed in this chapter, I'm giving a quick, very quick snippet or you can say snapshot of the chapter. So this is how you will also have to make notes. So you take a chapter, so you recollect whatever you have studied, write it in short so that you are able to recollect. That is how you have to make short notes. Okay, Satya? Yes. So this is your G1 phase. So S is your synthetic phase and this is your where your DNA replicates. So here DNA replicates but your chromosome number remains the same. So this is the most important thing and your histone proteins are synthesized here. So then you have your G2 phase. So in your G2 phase, so this is post DNA synthesis phase, okay. So this G2 is where your tubulin proteins for your microfibers are synthesized and your cell stores ATP, right? So these are some of the points. Yes, Vishal, hello, a very happy good evening. Thank you for joining my lecture. So please do like, share and subscribe. You thought the class is at 6:30. Okay, that's fine. Okay. So this is one part. So then we went on to talk about the most important part, that is mitosis. So mitosis is divided into prophase, metaphase, anaphase and telophase. So what happens in prophase? Yes, so from your chromatin, it condenses to form what is called as your chromosomes. Your cell organelles disappear, right? So this is what happens in your prophase. Yes, Vishal, I'm good, thank you. How are you? Okay, so this is your prophase. So here in prophase, so you have your centrioles which have duplicated in your interphase. So your centrioles begin to move towards the opposite poles. So centrioles, they have duplicated during the interphase. So they move towards the opposite poles, okay. So next you have your metaphase. So metaphase is where your chromosomes align in a particular equator and this is known as your metaphase plate. So metaphase is the stage where you have the best, where you can study the morphology of the chromosomes at its best. You have perfect shape and structure that is visible at your metaphase plate. Okay? Yes? What about physics and chemistry lectures? So as of now, physics and chemistry lectures are not happening in this channel. Yes, maybe very soon they will start, but I'm not sure about it. Yes, Anthony, yes, Prince, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. So this is about the first part. Then in anaphase, what happens? So your chromosomes segregate, right? So they segregate as chromatids. So you have arms of the chromosome. So here if I have a chromosome like this, so here they segregate like this. So with the help of spindle fibers, so spindle fibers attach to the disc-shaped structures known as kinetochores. Okay? Yes? Clear? So then you have telophase. So telophase is once again when your chromosome condenses to form chromatin. So this is just opposite to your prophase. So all the cell organelles are reformed. So this is what happens in your mitosis, okay? Yes, only if there is menti, it is at 6:30. So today we had a lecture, so today it was at 6, okay? Fine. So one more announcement. So tomorrow at 4 p.m. I'm starting human physiology. I'm starting with the first chapter, that is digestion and absorption. And tomorrow at 6:30 we are having guess the chapter series. So it is episode 9. So please be there. Okay. So this is about your mitosis. So why is mitosis important? So mitosis is important for cell repair. So mitosis is also important for your cytoplasmic to your nucleus ratio. To maintain this ratio also, your mitosis is important. The next type of cell division is your meiosis. So mitosis is known as equational division because a mother cell gives rise to two daughter cells with the same set of chromosomes. Yes, Keshav, hello, hi. So thank you for joining my lecture. So please do like, share and subscribe. Okay, but meiosis is your reductional division. So what happens in meiosis? So this is reductional division. So you have two types of meiosis, that is meiosis 1 and meiosis 2. So in meiosis 1 also prophase, metaphase, anaphase happens. So here also you have prophase, metaphase happens, right? But here the problem is prophase 1 of meiosis 1 itself has 5 substages. So what are the 5 substages? First is your leptotene, then it is your zygotene, then it is pachytene, then it is diplotene, then it is diakinesis. Okay, so this is how it is. So here, this is the normal prophase. So your chromatin condenses to form chromosomes. So that is what happens in your leptotene. So in your zygotene, so one chromosome from your father, one chromosome from your mother comes together. So this coming together of chromosome is known as synapsis. Okay, so if equivalent or homologous chromosomes come together, they are called as bivalent. So if for example, I say we have 46 chromosomes, which means you have two 21st chromosome, right? So you have one copy from your mother, one copy from your father, right? Yes, so I have the homologous chromosome coming together. So I call them as bivalents or tetrads. So if my total chromosome is 46, so my bivalence will be 23. Okay, clear? Fine. In pachytene, crossing over happens. So if this is a chromosome from mother and this is a chromosome from father, so this is where crossing over happens. So that is why you have a combination of both of them. Okay, so this crossing over place is supported by a synaptonemal complex. And this X-shaped structure is known as your chiasmata. So in your diplotene, this crossing over separates, so it is done crossing over. So you have new chromosomes that is present, that is crossed over chromosome. So in diakinesis, now you have terminalization because the chromosomes are ready for the metaphase stage. Okay, so in meiosis 1, at the end of meiosis 1, so you have two daughter cells present. You have two daughter cells. So and meiosis 2 is similar to your mitosis. So here each one of the two daughter cells undergoes meiosis 2. So at the end you have 4 haploid cells. So this is the process of meiosis. Is this clear? Yes. So this is your entire process of cell cycle and cell division. I hope it's clear. Yes. So can we go to the questions now? Ready? So before we go into the questions, so shall we set a timer? So for each question, I will give you 40 seconds, okay? So let's go with maximum time. So I'm not being stringent, okay? So I will give you 40 seconds for each question, but you will give me the answer. And you will self-evaluate how many you have got it right, so how many you have got it wrong, right? Okay, so all sad newses. Okay, fine. So here we go to question number 1. So shall I give you a time limit of 40 seconds? Is 40 seconds fine? So shall I set it to 40 seconds? Yes? So here we go. So today we will have it with the timer and see how it works. Okay? Okay, so your time starts now. So question number 1. So which of the following phase is called the resting phase, during which the cell is preparing for division by undergoing both your cell growth and the replication of DNA? Yes, quick. Yes. It is called the resting phase. Yes, the timer is here. I will pull it here. Yes. So, Prince, why does G0 phase? Interphase is also called as resting phase, right? Yes? Okay, fine. The time's up. Okay, so it has restarted. Okay, so the right answer is interphase. So this is your resting phase where the cell undergoes cell growth and replication. If the cell enters G0 phase, it will not proceed to the S phase. So interphase is one which has your G1, S and your G2 phase. Okay, clear? Fine. So here we go into the next question. Here I go. So question number 2. Which of the following phase follows S and G phases of the interphase? Which of the following phase follows S and G phases of the interphase? Yes. So I have G. So I have G1, S, G2. So I have M here. So M is prophase, metaphase, anaphase, telophase. Which of the following phase follows S phase and G phases of the interphase? Yes, what is your answer? Yes? Yes, come on, quick, time's up. Yes? What is the answer here? Yes, so prophase follows S and G phases of interphase, right? Okay, so that's absolutely right. So the right answer is prophase, right? Okay, so shall we move forward? Yes? Okay, so thank you, Spare Akash. So please do like, share and subscribe. Okay? Yes, so here we go to question number 3. So shall we go? Yes, so once again the timer is set at 40. So which of the following cellular structures always disappears during mitosis and meiosis? Plastids and nuclear membrane, nucleolus and nuclear membrane, endoplasmic reticulum and mitochondria, endoplasmic reticulum and plasma membrane. Yes, what is the answer? Yes, Prince, you have given me the right answer in the quickest time. Absolutely brilliant. Yes. Yes, so all of you have given me the right answer. So your nucleus and your nuclear membrane, they always disappear, because if this is your nucleus, inside your nucleus you have nucleolus and surrounding it you have your nuclear membrane, right? So this first disintegrates. Yes, absolutely right. So it is it is option B, right? So here we move forward to question number 4. So in meiosis, division is first reductional and then equational, or first equational and then reductional, both reductional, both equational. Yes, what is your answer? Yes. First reductional and then equational. Yes, absolutely right. Keshav, Prince, Shimpi. In meiosis, I am asking. So I will pause. So in meiosis, So I have meiosis 1 and I have meiosis 2, right? So in meiosis 1, what happens? I have a mother cell who's 2n. She gives birth to daughter cells who are just n, right? Because here you have crossing over. So the two chromosomes separate from each other, right? Okay. So now in meiosis 2, what happens? I'm talking about this n. So this n gives me two daughter cells of the same type. So this n gives me two daughter cells. That is why at the end of meiosis, I have 4 cells, right? So it is first reductional division, it is half. Next one is equational division. So the right answer is option A. Okay? Okay, so here we go to the next question, question number 5. So synaptonemal complex appears between homologous chromosome in zygotine stage composed of DNA plus proteins, all of the above. What is your answer? Yes, what is your answer? Yes, Keshav, absolutely brilliant. So you have given me the right answer in almost 11 seconds. Very good. Yes, Prince, it is D. Yes, Shimpi also absolutely right. So fantastic. So you guys have given me the right answer. Okay. So synaptonemal complex is the one that confers stability at the time of crossing over. So it is between homologous chromosomes. So what are homologous chromosomes? Chromosomes of the same type. Suppose if I talk about chromosome 6. So I have 2 copies of chromosome 6. So these two, one from father, one from mother. So this is my homologous chromosome. So this happens in the zygotine stage of my prophase 1 of my meiosis 1. Right? So it is composed of DNA plus protein and all of the above here is right. So this is option D. Yes, so here I move forward to question number 6. So chiasmata are first seen in leptotene, zygotine, pachytene, diplotene. Yes, take your time. So chiasmata are first seen in which of the following? Yes, quick. So it's pachytene, diplotene. Okay, so chiasmata is your crossed over portion, right? So in pachytene is where crossing over happens, okay? Okay. So leptotene is the first stage. So here you have nothing. So here your chromatids become your chromosomes. Fine. So the next one is your zygotine stage. So in zygotine is where you have synapses. So in synapses, the chromosomes are getting ready to be crossed over. So here you have in the formation of bivalents and your tetrads. Right, so next you have pachytene. So pachytene is where you have crossing over. So crossing over is happening. So in diplotene, my crossed over chromosome is like this. Right, so chiasmata is this X-shaped portion. So before this, it is like this, right? So the chromosomes try to separate from each other except at this position. So this is my chiasmata. It is first seen in my diplotene phase. So chiasmata is a portion that is present after crossover. Okay? Yes? Yes, after crossover it will happen. So the answer is diplotene here. Fine? So shall we go to question number 7? So totally itself I have 20 questions. So we are going at a much faster rate. So splendid. Question 7. So, in which stage chromosomes appear thin and long thread-like structures? Zygotene, leptotene, pachytene, prophase. Yes, chromosomes appear thin and long thread-like structure. Yes? Okay, so your Keshav from your phone account. Okay, Keshav, so please do like from your phone account as well. So, in leptotene stage. So, why not in prophase stage? Okay, so you all have got the concept right. So yes, the right answer is leptotene. So perfect. So leptotin is the first stage of your prophase 1. So here from thread-shaped chromatin, it becomes condensed as chromosomes. Yes, absolutely perfect. So very good, guys. Okay. So moving forward to the next question. So here I reset the timing. So electron micrographs of which stage indicate that chromosomes accompanied by the formation of complex structure called as synaptonemal complex? Zygotine, pachytene, diplotene, diakinesis. Yes? Wow, thank you so much, Keshav. So very good. So diplotene is where you see synaptonemal complex. Yes? So synaptonemal complex formation happens in which phase? Yes, ma'am. A very, very happy good evening. So thank you for joining my lecture today. So please do like, share and subscribe. Yes. So synaptonemal complex is seen at the time of crossing over. So it is during the pachytene stage. It is accompanied by the formation of complex structures called the synaptonemal complex at pachytene stage. No, zygotine is where you have synapses. The chromosomes come together. They just get ready. Okay, Prince. So here the right answer is option B. Fine. Okay, so very good. So you guys have given me the answer in just 20 minutes. So perfect. So question number 9. So mark the incorrect pair. Mark the incorrect pair. So interphase is S phase, M phase is metaphase, meiosis 1 diplotene, meiosis 2 is diakinesis. Yes, I want the incorrect pair. Interphase is S phase, so M phase is mitotic phase. Yes, you can say metaphase as well. So in meiosis 1, you have diplotene, but meiosis 2, you do not have diakinesis, right? So meiosis 2 is similar to your mitosis. So here you do not have diakinesis stage. It is this prophase, metaphase, anaphase. So this is the incorrect pair. Yes, brilliant. Great. Okay. So yes, Shambhavi. So thank you for joining my lecture. So please do like, share and subscribe. So here we move forward to the next question. How many mitotic divisions are needed for a single cell to make 128 cells? So all of us know one mother cell gives rise to 2 daughter cells in a mitotic division, right? So how many mitotic divisions I have to totally make 128 cells? So one cell gives me 2 daughter cells. So it is 2 power 1. So 2 power n should give me 128 cells. Now what is n? Yes, so Prince says 7. Yes, that's absolutely right. So yes, your time is up. Yes, so only Prince has given me the answer. Vishal says A. Okay, so Shimpi, what is your answer? Antonia B. Dharni, what is your answer? No, n minus 1 will not come here. So it is 2 power n gives you the number of divisions. So 2 power 1, so is 2. Right, so 1 is the 1 division, 1 meiotic division, it gives me 2 cells. So if it's 2, it's 2 power 2, that is 4, right? Yes, so this is 7, so 2 power 7 is 128. So I have to undergo 7 mitotic division of my mother cell to give me 128 cells. Yes, absolutely right. So here I move forward to question 11. So how many meiotic divisions are required to produce 100 pollen grains? 25, 50, 100, 125. Yes, your times are happening. So let's see who gives me the right answer first. Yes, Keshav has given me the right answer in just 15 seconds. Yes, brilliant. So followed by Prince. Yes, Shimpi, so this should not be tough. Very good, Shimpi. Perfectly right. So good. So my kids have given me the right answer. Okay. So how it is 100 pollen grains? So if I have a pollen mother cell, so it undergoes, because in one meiotic division, so in the process of meiosis, which includes both meiosis 1 and 2, so at the end, I have 4 daughter cells, right? So I have 4 haploid cells. So my pollen grains are haploid. So if one meiotic division can give me 4 cells, right? So then 100 pollen grains I need. So how many divisions it will be? So it is 100 by 4, that is 25 divisions. Yes? Yes, great, very good. So Ranjan Maheshwari, yes, thank you for joining my lecture today. So please do like, share and subscribe, okay? So for 200 pollen grains, how many will you need? 50 you will need. Yes? So you have 25 divisions for 100, right? So for 200, how much it will be? So this into 2. So this also into 2. So it will be 50. Hannah Vishal? Yes? Okay, so here the answer is option A. Good, great. So question number 12. If you are provided with the root tips of onions in your class and you're asked to count the chromosome, which of the following stages can you most conveniently look into? Metaphase, telophase, anaphase, prophase. So only when the chromosome, for seeds we need to, yes, you're right, Vishal, yes. So in which stage will you look into the structure or you will count the chromosomes? So in which phase will the chromosome be very visible? Yes, absolutely right. So this is metaphase. So metaphase is used to study the morphology of chromosomes. It is also used to count your chromosomes. Yes, perfect. So option A is the right answer. Okay, so here we go into question. No, not anaphase, it is metaphase because chromosomes align properly in a particular equator at metaphase. So in this metaphase only you can see clearly see the structure of your chromosome. Okay, Ranjan, so the right answer is option A. Fine. Okay, so moving forward. So question 13, the number of bivalents are 8 in prophase 1. What is the number of chromosomes during anaphase 2? Yes, quick. Yes. So I want the number of chromosomes during anaphase 2, that is meiosis 2. Yes, quick. Come on, give me the right answer. Last few questions. So I have only 20 questions. Shimpi, 32. How did 32 come? Do you like the number 32 very much? Yes, Vishal, absolutely right. It is 8. Very good. Shambhavi says 32. No. I'm asking the chromosomes, not the amount of DNA. Yes, it is option A. How? So number of bivalents. So bivalents is 1 chromosome from your mother, 1 chromosome from your father. If 2n is 46, then bivalents will be n, right? So this is my bivalent or my tetrad. Clear? Yes? Okay. In one class you said, ma'am, I asked this. Okay, so this is option A. Yes, you're right. Yes, Swami, very happy good evening. So thank you for joining my lecture. So guys, come on, please do like, share and subscribe the session. So N is 23. So this denotes the bivalents and the tetrads. Okay. So here after crossing over, my chromosomes separate. Right? So initially if I had 2n, so assume my 2n is 16. So the number of bivalents is 8. Right? In my anaphase 2, I am going to have only haploid cells. Right? Yes, anaphase 2, I am going to have haploid cells because at the end of meiosis 2, I am going to have haploid cells. So I will have only n number of cells. Right? So it is 8. It is 8. They are asking about chromosome only. No, Shimpi, it's pretty clear. So whenever they give you the bivalent, so that denotes n. So the organism is 2n. Okay. So the number of bivalents will be the number of daughter chromosomes. Suppose if I have a mother cell who is 2n. So she is undergoing meiosis. So at the end of meiosis 1 and 2, she will give me 4 cells, right? So 4 cells will be haploid. So if initially she is 16, so this will be 8, 8, 8. So maybe because of which you said 32, but they are asking me only in anaphase 2 of 1 cell. They have not mentioned in total. Okay, so now I get how you said 32, but it will be 8. They are asking about a single cell. Alright, clear? Fine. So here we move forward to question number 14. So in telophase of mitosis, the mitotic spindle breaks down and the nuclear membrane reforms. This is essentially the opposite of what happens in prophase, interphase, metaphase, S phase. So I told you, so telophase is exactly opposite to one of the phase. Which phase is that? Yes, quick. Yes, quick. Yes, absolutely right. So it is prophase. Because in prophase, what happens? The mitotic spindle tries to form, the nuclear membrane disintegrates, right? So here everything happens in the reverse way. Yes, here it is prophase. Absolutely right. So question number, okay, so 14 is done. So we are going to question number 15. So X ensures the production of Y phase in the life cycle of sexually reproducing organism, whereas fertilization restores the Z phase. So identify X, Y, Z. Yes? So if whenever I say fertilization, so fertilization is the fusion of your sperm and the egg. Right? So sperm is haploid, the egg is haploid, so they restore the diploid phase. Yes? So here the Y phase should be haploid and this should be meiosis. So the right answer here is option C. Yes. Yes. Yes, absolutely right. Okay, so perfect. Yes? So question number 16. 4 daughter cells formed after meiosis are genetically similar, genetically different, anucleate or multinucleate. Yes, 4 daughter cells formed after meiosis are genetically similar, genetically different, anucleate or multinucleate. They are genetically different. 4 daughter cells formed after meiosis are genetically different. They're genetically similar, right? Because they come from the same mother cell. Why this doubt? Why this doubt? So I have one mother cell 2n. So she is undergoing meiosis 1. So she gives me 2 daughter cells. Now this is undergoing meiosis 2. So this gives me equational division. So it is genetically similar, right? Yes? 2-2, what is 2-2? How can it be genetically different? Yes? Okay, so I have a father 2n, he's trying to form gametes. So the gametes are XY. So this X is similar to another man, this Y is similar to another man. No, right? They are similar to a same parent. Yes, they are genetically similar but morphologically different. Yes, that is the right point. Okay? Yes. So here we go into the next question. So here, I'm sorry, yes. So in animal cells, cytokinesis involves the separation of sister chromatids, the contraction of the contractile ring of microfilament, depolymerization of kinetochore microtubules, the protein kinase that phosphorylates other enzyme. Yes, the contraction of the contractile ring. So in animal cells, it is cell furrow formation that happens through your contractile ring. Yes, perfectly right. Very good. So please don't note in how many seconds you are able to give me the right answer. That is also important. So here we go into question number 8. Ready? So if a tissue has at a given time 1024 cells, how many cycles of mitosis had the original parent cell undergone? Yes, quick. So one cell gives me 2 daughter cells, right? So it's 2 power 1, that is 2. So 2 power n is the formula. So how many divisions it will give me? 1024 cells. Yes, 10. So 2 power n. So n will be 10. So 2 power 10 is 1024. Right? So I hope you remember the shortcut of learning how to find out the squares. So 2 power 10, how will you do? So you write the same number, the number here and twice this, right? So I hope you remember the short trick if you have read. So this is how in a short way you can find out. Okay. Cousins are fighting, but Prince is fastly answering. Oh, okay, yes. So here we go to question number 19. A cell's division time is 1 minute, okay? In 1 minute, a cell completely divides. In 20 minutes, a culture tube is 1/8 filled with cells. When the tube will be fully filled? Yes, give me the answer. Yes, give me the right answer for this. How quickly are you giving? Let me see. Come on, quick. Yes, come on, quick. D, 160 minutes. Oh my God, Shimpi. The total time of cell division itself is 1 minute. 160 is too much. Yes, Keshav, B is the right answer. Okay, so let's see how B is the right answer. So, in 1/8, so in 20 minutes, 1/8 is filled, whatever it is, plate or square or something. In 20 minutes, 1/8 is filled. So if I take the next minute, so in 1 minute, my cells divide, right? So I have 1/8 cells. So each cell divides into 2, right? So in 20 minutes, 1/8 cells are there. So in the next 1 minute, these cells will divide. So each cell will give me 2 daughter cells. So I will have 1/4 cells, right? So in the 22nd minute, so 1/4 will also divide. So this will give me 1/2 cells, right? Yes. So in my 23rd minute, so 1/2 also will divide. So that will give me a whole number, which means the entire media is filled. Right? Yes. So this is 23 minutes. So that is the right answer. So did everybody understand? Okay. So 1 minute is the time for the cell to divide. So in 20 minutes, 1/8 is filled. So, okay. So in 21 minutes, so this 1/8 will divide, right? So one cell will give me 2 cells. So 1/8 into 2. In the next 1 minute, 1/4 will divide into 2. So it is 1/2. So in the next minute, 1/2 into 2, that is a whole number, which means the entire plate is filled. Clear? Yes? Yes? Yes, Keshav, you're also right. Okay, so this is question number 19. That is answer is option B. So these are the questions which you might face difficulty, but as such, this chapter is pretty easy. Okay, so I hope you have understood. So here we go, the last question for the evening. So a spindle usually persists in the form of dash during dash method of cytokinesis. Yes, quick. A spindle usually persists in the form of dash during dash method of cytokinesis. Yes, quick. Yes. So what is phragmoplast? Yes, time's going. Yes, Shimpi, absolutely right answer. Yes, Prince, good. Yes, Antonia, be right. Okay, so yes, Shambhavi, right, you're perfectly right. Okay, so this is phragmoplast. So phragmoplast is the formation where your Golgi complex comes and aligns at a particular point, and this is in plants. So in plants, it is cell plate method. So here the right answer is option B. So these are some of the tricky questions where you tend to make mistake. So as such, this chapter is pretty easy. So I have completed this unit. Okay, so 17/20. Okay, but it took time. Okay, fine. See, though it's an easy chapter, so you have lost 3 questions, right? So please pay a little attention. So I am done with this unit, that is cell, the unit of life, biomolecules, cell cycle and cell division, right? So you will have a mega menti on Friday or Saturday. So please prepare these 3 units. I am informing you well in advance. Yes? Okay? So please don't forget to join me tomorrow at 4 p.m. for human physiology. I am starting the first chapter. Shambhavi, it is at 4 p.m. tomorrow. So at 4 p.m. we are starting human physiology, that is digestion and absorption. So tomorrow at 6:30 we have a Menti quiz. So that is episode 9. So it is just the chapter Menti series. Okay? Yes? So thank you so much for joining my session today. So if you genuinely liked my way of teaching, so please do like, share and subscribe. So this is me wishing you more and more. So thank you and looking forward to meet you tomorrow at 4 p.m. So please join me for the same and have a great evening ahead. Bye bye and take care.